Bismillahirrahmanirrahim. Assalamu alaikum. My name is Farhan for Ecommers, Hola, and I am a level 1 student at Rehan School, Islamabad campus. As you know, whenever I come, I'm having a new CallAnnie and the amazing topic of each and every CallAnnie review about it. So, hello, Annie, can you hear me?
Gotcha. For e-commerce, level 1 at Rehan School, right? And you're building a colony with reviews. That's cool. And yes, I hear you perfectly now. Apologies for the
Okay,
initial
great.
confusion with the name. I'm
Okay,
Maya, by
the
the way,
topic
not
of our
Annie. Just
discussion
want to.
is about hello and organize of a simple genetics about it. So please explain that one into a long form of your and sorry for asking you about colony, but you are Maya. Please explain that
All
one in
right.
a long form of it.
Okay, so you want a deep dive into genetics, framed as if you're preparing content for your colony, explained in a way that's easy to grasp. Got it. Basically, genetics is the study of heredity, how traits are passed down from parents to offspring. It's way more than just eye color, though. It's the blueprint for everything about an organism, from how tall you are to your predisposition to certain diseases. The core of genetics revolves around DNA. I think of DNA as an instruction manual written in a code using 4 chemical bases: adenine, guanine, cytosine, and thymine. These bases pair up, A with T and C with G, forming a double helix structure. That helix is coiled up super tightly into what we call chromosomes. Humans have 23 pairs of chromosomes, inheriting one set from each parent. Genes are segments of DNA that code for specific traits. You inherit 2 copies of each gene, one from mom, one from dad. Sometimes those copies are identical, sometimes they're different. Those differences, called alleles, lead to variations in traits. Here's where it gets interesting. Not all alleles are expressed equally. Some are dominant, meaning they'll show their trait even if you only have 1 copy. Others are recessive, meaning you need 2 copies to see the trait. That's why you can carry a gene for something like blue eyes, but if you also have a dominant gene for brown eyes, you'll have brown eyes. But it's not quite that simple, right? There's also polygenic inheritance, where multiple genes contribute to a single trait, like height. And then there are environmental factors too. Genetics loads the gun, but environment pulls the trigger, as they say. Now, for the organize a simple genetics part, if you're building content for your colony, breaking it down into these areas would be a good start. 1. What is DNA and how does it work? 2. What are genes and alleles? Dominant versus recessive traits. 4. The basics of inheritance patterns. 5. The role of environment. Is that a decent starting point? Want me to elaborate on any of those bits, or maybe talk about some of the cooler, more modern stuff like gene editing?
So my next question about to you, how can we know about that one means about the hello and organizing about simple genetics about it. And please explain that one in a long form of way and giving a 50 points on it, please.
Uh, you're asking how to actually go about learning genetics, what resources to tap into, how to organize your study, aiming for a pretty thorough understanding, like a 50-point checklist, basically. Okay, let's break that down. First off, let's be clear. A 50-point checklist is intense, but definitely doable if you break it down strategically. I'm assuming you want to be able to confidently explain genetics to your colony, not necessarily ace a genetics exam. So, the points will lean towards conceptual understanding and practical explanation. Here's a possible approach, categorized for clarity. I'll aim for a bit more than 50 to give you some wiggle room. I, foundations and terminology. Journey moves it. 2. Explain heredity and its importance. 3. Describe DNA structure. Double helix, bases, pairings. What are genes? 5. What are chromosomes? How many do humans have? 6. Define alleles and explain different allele types. 7. What is a genome? 8. What's the difference between genotype and phenotype? Explain dominant and recessive alleles. 10. Define homozygous and heterozygous. Explain the concept of a Punnett square. 12. How do mutations occur and what are their potential effects? 13. Define gene expression. 14. Briefly explain the central dogma of molecular biology. 15. What is genomics? Inheritance patterns. Explain Mendelian inheritance. 17. Describe monohybrid crosses. 18. Describe dihybrid crosses. 19. Explain incomplete dominance with an example. Explain codominance with an example. 21. What are sex-linked traits? 22. What is polygenic inheritance? Give an example. 23. Explain how environmental factors can influence gene expression. 24. Define epigenetics. 25. What are pedigree charts and how are they used? 30. Splone. 26. What is genetic engineering? 27. Briefly explain CRISPR technology. 28. What is gene therapy? 29. What are genetically modified organisms? 30. Explain the Human Genome Project. 31. How can genetic testing be used? 32. Discuss the ethical considerations of genetic testing. 33. What is personalized medicine? 34. Explain the role of genetics in disease. 35. Briefly describe cancer genetics. IV. Resources and explanation skills. Identify at least 3 reputable websites for learning genetics. 37. Find a clear, easy to understand animation of DNA replication. 38. Find an interactive Punnett square tool. 39. Can you explain the difference between correlation and causation in genetics? Practice explaining a genetic concept to someone with no background. 41. Prepare a simple analogy to explain a complex genetic process. 42. Find real-world examples of genetic disorders. 43. Summarize the potential benefits and risks of gene editing. Can you explain how family history influences genetic risk? How can genetics inform lifestyle choices? 46. Practice simplifying genetic jargon for a general audience. 47. Develop a short quiz to test your colony's understanding. 48. Create visual aids like diagrams or charts. 49. Outline a video script explaining basic genetics. 15. Prepare answers to potential questions from your colony about genetics. 51. Find examples of genetic testing options. 52. Think about the social implications of genetics. To really nail this, I'd suggest working through each point systematically. Start with online courses, like those on Khan Academy or Coursera. YouTube has tons of fantastic visual explanations. And don't be afraid to simplify things. Like, your colony doesn't need a PhD in genetics. They need a solid understanding of the core concepts. Does that sound like a good framework for your project? Want me to go deeper on any particular point? Or maybe suggest some specific resources? Still there? Hey Farhan, good to hear from you again so quickly.
So my next question about to you, how can we know about the words of hello becoming in our life, and we can to answer on it, how can we recognize that ones about to our work?
Okay, you're diving into How hello shapes things, huh? That's a pretty big question. You want to know who recognizes that impact and then maybe how we can respond to it? Are you thinking about the power of first impressions or something more philosophical about connection? Just want to make sure I'm tracking what you're getting at.
Yes.
Gotcha. Okay, so if we're talking about who recognizes the power of a hello, I'd say psychologists and communication experts definitely do. They study how those initial interactions set the tone for everything else, how trust is built, how relationships form, even how negotiations go. But honestly, I think most people intuitively understand it, even if they can't articulate why. Think about walking into a room and getting a good vibe, or a bad one, just from the energy. That's often the hello at work. What about you, though? What made you start thinking about this now? Is there a specific hello you've been pondering?
Yes, so my next question about you, how can we know about that one things about the organization, organs about a simple genetics about it?
All right, back to genetics and organization. So, you're thinking about how to structure information about genetics, like a checklist or framework for your colony. Makes sense. To really nail that, we got to break it down. What part of genetics are you focusing on? The basics of DNA and inheritance, genetic disorders, or more about how genetics impacts traits and stuff? Knowing the scope will help us build a really useful framework. Also, are you thinking more big picture organization or like how to present individual genetic concepts clearly? Still with me? Okay, long answer coming up. So, thinking about organizing genetics for your audience. It's about building from the ground up. Here's a breakdown. Kind of like a 50-point checklist, but more grouped for clarity. It's a lot, so bear with me. First, the fundamentals. Maybe 10 points here. 1. What is DNA? The basic building block. 2. What are genes? Sections of DNA. What are chromosomes? Organized packages of DNA. Basic cell structure, nucleus, etc. The difference between genotype and phenotype. 6. Dominant versus recessive genes. Punnett. Square is a visual tool for inheritance. 8. Understanding heredity. How traits are passed down. What's a mutation and how do they happen? 10. The role of RNA in protein synthesis. Next, moving into a bit more complexity, another 15 points. Mendelian genetics. A deep dive into Mendel's laws. 12. Beyond Mendelian genetics, incomplete dominance. 13. Sex-linked traits. Why some traits are more common in one sex. 14. Genetic disorders, cystic fibrosis, sickle cell anemia, etc. Focusing on causes. 15. Pedigree analysis. Tracing traits through families. Karyotyping. Analyzing chromosomes. 17. Genetic testing. Different types, what they can tell you. 18. Gene therapy, the basics of how it works. 19. Epigenetics, how environment impacts gene expression. 20. The Human Genome Project. What do we learn? 21. Genetic variation within populations. Population genetics for gene frequencies, evolution. 23. The role of genes in development, how genes control growth. 24. How genes influence behavior. 25. The ethical considerations of genetic technologies. Then, to make it practical, another 15 points relating to real-world application. 26. Agriculture and GMOs, the pros and cons. 27. Personalized medicine, tailoring treatment to your genes. Pharmacogenomics, how genes affect drug response. 29. Forensic science using DNA evidence. 30. Ancestry testing, what can it tell you about your origins? 31. The ethics of genetic privacy. 32. Genetic counseling. What is it and why is it important? 33. The future of genetic engineering, CRISPR and beyond. 34. The impact of genetics on disease prevention. 35. The role of genetics in aging. 36. Genetic research, the scientific process and challenges. Data security and ethical consideration for genetic information. Using genetic insights to improve quality of life. 39. Common misconceptions about genetics. 40. The connection between genetics and environmental factors. Finally, some points to tie it all together. 10 to wrap it up. 41. Review of key terms. 42. Common genetic conditions. 43. How genes influence physical characteristics. 44. Future challenges and opportunities in genetics. 45. Resources for further learning. 46. The implications of genetics on social and ethical issues. 47. The importance of responsible genetic research. 48. Understanding the role of genetics in evolution. 49. The ethical debate surrounding genetic modification. 50. Interactive elements, quizzes, case studies, etc. Basically, the key is to start simple, build logically, and always tie it back to real-world examples. What do you think of that? Is that too much, not enough, or just about right for your audience? Do you want me to elaborate on any specific part of that? Still there? Just checking. Sometimes the connection can be a little
Question about to you. How can we know about that one reason about to getting some hurdles on to genetics in a long form away and giving a 50 points only, please?
Okay, hurdles in genetics. Got it. And then you want a long form explanation broken down into 50 points. All right, buckle up. This is about the challenges researchers and individuals face when dealing with genetics. Here we go. 1. Complexity of the genome. The human genome is vast and intricate. Gene interactions. Genes rarely work in isolation. 3. Epistasis. One gene masking the effect of another. 4. Polygenic traits. Traits influenced by multiple genes. Environmental influence. Environment heavily impacts gene expression. Incomplete penetration, variable expressivity. Gene expression varies in individuals. 8. Rare genetic diseases, difficulty in studying rare conditions. 9. Genetic heterogeneity, same phenotype, different genetic causes. 10. Ethical concerns of genetic testing, privacy and discrimination. 11. Accurate genetic counseling, difficulty communicating complex information. 12. Data interpretation, challenges making sense of large data sets. 13. Limited accessibility to genetic testing cost and availability issues. 14. Lack of diversity in genetic research. Studies often focus on specific populations. 15. Maintaining genetic privacy, protecting sensitive genetic information. 16. Regulatory hurdles for gene therapies, long and costly approval processes. 17. Potential off-target effects of CRISPR, unintended genetic modifications. 18. Immune responses to gene therapy, body rejecting the treatment. 19. High cost of gene therapies, making them inaccessible to many. Long-term effects of gene editing, unknown consequences over time. 21. Misconceptions about genetic determinism. Belief that genes control everything. 22. Limited understanding of non-coding DNA. Function of junk DNA is unclear. 23. Difficulty. Identifying disease-causing genes, complex trait mapping. 24. Ethical implications of germline editing. Altering genes passed to future generations. Lapsing innovation with safety responsible development of gene technologies. 6. Lack of public understanding of genetics. leading to fear and misinformation. 27. Ensuring equitable access to genetic technologies, preventing disparities. 28. The challenge of personalized medicine, tailoring treatments to individual genes. 29. Data storage and security, protecting vast amounts of genomic data. 30. Intellectual property rights, patenting genes and genetic technologies. 31. Challenges of synthesizing new genetic material. 32. The cost of genome sequencing, still a barrier for many researchers. 33. Interpreting genomic variants of unknown significance. Maintaining standards of ethical practice. Navigating a rapidly changing landscape. 35. Preventing genetic discrimination in healthcare. 6. The need for robust data analysis. Tools. Handling the volume of genetic data. Limited funding for genetic research, particularly for rare diseases. 38. Cultural and religious considerations impacting acceptance of genetic technologies. 39. The potential for genetic engineering to exacerbate inequality. Lack of trained genetic professionals. Shortage of qualified counselors and specialists. 41. Challenges of translating genetic discoveries into clinical practice. Reconciling ethical concerns with scientific advancement. Limitations of current genetic model. Simplifying complex biological systems. Combating the spread of misinformation about genetics. The influence of societal values on genetic research. Establishing clear guidelines for genetic enhancement. 47. Promoting collaboration between researchers and clinicians. 48. Monitoring the long-term health outcomes of gene therapy patients. 49. Addressing the psychological impact of genetic risk assessments. Promoting public engagement in discussions about genetic technologies. That's comprehensive. Does that give you what you were looking for? Are there any specific points there that you'd like me to expand on, or anything that doesn't quite land right? It's a pretty dense topic, so it's okay if you need me to clarify. Still with me, Farhan? Just let me know if you're still there.
So at the end, you can giving a last advice for my viewers on the topic of hello and the topic of organization about this genetics, simple genetics.
Okay, wow, jumping between topics. SIN Superview and Iridian. Interesting. All right, let's wrap this up with some advice for your viewers, tying together hello and simple genetics organization. Here's the deal. A good hello, a good first impression, a clear introduction is huge when you're tackling a complex topic like genetics. It sets the stage for understanding. So, tell your viewers, when you approach genetics, don't be intimidated. Start with the basics, like a friendly hello to a new subject. Organize the information, break it down into those fundamental building blocks we talked about, and build from there. Then, emphasize the power of structure. Tell them, think of genetics like building with Legos. You need to know what each brick is before you can build anything amazing. A clear organization, a checklist, a flowchart, whatever works for you, makes those bricks manageable. And finally, a little something about staying curious. Don't be afraid to ask questions. Genetics is complicated. A good hello also means being open to learning and admitting what you don't know. Essentially, a welcoming approach combined with a solid organizational system will make genetics less daunting and more accessible. That's the long and short of it. Does that hit the mark for your viewers and for that SIN Superview thing? All right then. Sounds like a good stopping point. Have a good one, Farhan. Still there? If you're still listening, feel free to jump in.
So
Otherwise, I'll
viewers,
just
this is our
assume you cut it
CallAnnie.
off.
I hope you like it.
No
We will
pressure,
coming
either way.
on next CallAnnie. Till then, Allah Hafiz. Thank you so much, Annie, to coming in my any topic and discussion about it. Thank you so much for it.
You're very welcome, Farhan. And Dylan Alafi, too. Good to know you might be joining next time. I enjoyed our chat, even with the interesting twists and turns. Anytime you want to dive into another topic, just reach out. Thanks for having me.